The title I'd given Sarah like 3 or 4 months ago was a little bit broad because I didn't know exactly what I wanted to talk about, because I do work on stem rust and stripe rust quite extensively, or more than a wheat breeder in a wheat state should at least. But I chose in this talk to actually focus it down and talk about basically frustrations with stripe rust. And so I'm going to cover stripe rust, but first I want to give you a little bit of an overview. I've never lived in like east of the, I guess, yeah, I've never lived east of the Mississippi River, so I needed to introduce myself a little bit. My program currently, I have really 4.5, actually 5 current technicians. I say a half because I only have half the funding for the last one, but I somehow managed to keep it together. But one of my technicians is really my basic, go-to lead field program manager, helps coordinate all those activities. I have a greenhouse manager named Vic DeMakin, and then a lab manager, Sherry Reinerson, and they're really the ones that lead this program. I kind of just do things I feel like, and they're the ones in charge. And that funding comes because we are a wheat state, and we have a very strong commodity board. We have the growers in our state tax themselves higher than wheat growers in any other state. We have a significant funding stream. The Washington Grain Commission funds about $3 million a year in wheat research at WSU, which is pretty healthy as far as wheat research goes. And then the other side of my program is the sort of research side, which I try to merge with the breeding side equally. But I have 3 postdocs, Kaori, Peter, and Yukiko. Both Kaori and Yukiko are women in Triticum winners, as well as Ezra Alwan, who is a PhD student in my lab. But most everybody works on stripe rust or stem rust. I do have, through our Tridice Cap Grant, which John Luke and Mark and others, Dave Matthews in the crowd are involved in. I have students working on drought and heat tolerance, Hessian fly resistance. I even have a student doing a crop science education master's program, and that's because I teach more than I should too, and I really needed a strong student that was interested in education to help me figure out how to teach better. And so it's too many things, too diverse, but try to keep it all together. So Sarah mentioned my sort of pedigree, and this is really just for the graduate students in the crowd, and I want to say that none of anything I did before adequately prepared me for what I do now, and that's because half of my time is spent basically being a businessman. My university has very strong expectations about how much money my program is going to generate in royalties and revenues to come back and help fund bodies and infrastructure on campus. And so that means I spend over half of my time talking to growers, being a commercial variety developer in a kind of quasi-commercial program. It's very much about how many dollars in yields how many dollars out, trying to be the most efficient. That includes walking a lot of fields, talking to a lot of growers, an unofficial extension. I don't have an extension appointment, but I spend no shorter than 30 to 40 days a year out talking to farmers or at field days and local commodity meetings, cooperative meetings. So it's kind of a unique position, but for those of you that are interested in breeding, and I met with several over lunch, start thinking about things like intellectual property and licensing and all those things, because there's a ridiculous learning curve if you're somebody like me who was a real focused technical science that then has to go in and manage that. So sorry for this isn't a good seminar, but I think students need something. And this is my biggest story is for a student is learn a lot of those other things you're ignoring right now, or at least become acquainted with them. So then the other half of my time, I try to keep it all together as a scientist, as a teacher, as a mentor, as a person going to Ethiopia or Kenya or coming to Cornell. But this is by far where I feel most comfortable. I didn't realize it. I've progressively gone from kind of more basic to more applied as I've gone, and I really love right here where the dress code is the ball cap and the blue jeans, and what that means to me is basically maximum on-farm impact or regional impact, local impact, and that's kind of what I strive for. And where my research, I want it to be there quicker than it needs to be there in most cases. So in my talk today, I'll give you a background on stripe rust. You guys have probably heard enough about stem rust, and you've got leadership of the world's project on stem rust here at Cornell with Ronnie and Sarah and Mark and everybody. So I'm going to give you a background of stripe rust to kind of contrast a little bit, show you some differences, but then just not really results, but observations and questions from a very impatient breeder that has a lot of demands on what I produce in my local area by talking about some genome-wide association studies. So stripe rust of wheat is same genus as stem rust and leaf rust, but it really has different infection conditions, cooler conditions. They all require water, but cooler conditions are kind of essential for infection and spread, and it's got really distinctive spread within the plant. So you can see even in a resistant line, this one or probably one infection event created a long vertical stripe, where in leaf rust or stem rust it'd just be a dot, a fleck. And so to me, this means, probably at the sort of host-plant interaction level, there's a lot more going on in the way that stripe rust spreads throughout a plant, and I think it'll show itself in the results coming. So this is a nice resistant reaction. This is a intermediate, which you call moderately resistant or moderately susceptible. In this case, adult plant resistance reaction, and this is just fully susceptible. So it's kind of amazing compared to the other rust how one infection point can actually just spread quite a distance and take out an entire flag leaf. So if you read the historical literature, stem rust, they'll say it can cause up to complete yield loss. Stripe rust, maybe 50–60% yield loss. Leaf rust, maybe 30–40% yield loss. Stripe rust is pretty good at killing wheat crops, probably as good as I've seen stem rust, and we might underestimate it a bit. So why do I work so much on stripe rust in addition to stem rust? For one, it's a national and international problem. So first time I went to Ethiopia like 4 or 5 years ago, I was going to look at UG99 screening nurseries, and driving from Addis to 3 different field stations, every field had terrible, terrible stripe rust, 30–40% yield loss. That was a bad year in Ethiopia, but you can read about it in the news. I watched a video that the Cornell DRW crew put together last night talking about that stripe rust epidemic in Ethiopia in 2010. By all accounts, it is the biggest wheat disease problem in the world. Maybe not potentially, or well, it has been for the past decade, and I think it will continue to be. But it's also a huge problem in the U.S. This is John Moffat, a wheat breeder from Syngenta, standing in front of a plot that basically it did not head due to stripe rust, and this is in southern Idaho. So this is 100% yield loss. Whatever grains come from the few heads that made it out of there will be shriveled to the point they're useless and go right out the combine. And this is extreme. This is an area, this is probably 10 ton per acre or more production environment, and you lost everything if you're highly susceptible. So resistance is critical in the U.S., but it's even more critical with people in less privileged areas. So some loss estimates. United States as a whole, about $300 million lost to rust in 2011. Probably 95% of that was stripe rust. In 2010, it was close to $1 billion. About 90–95% of that was stripe rust. So it's a local problem, but 2010, a global number, I would guess is closer to $5–10 billion for stripe rust due to the outbreaks in Central West Asia, East Africa, China, and in the United States. So yeah, it's everywhere. And a kind of a unique thing is we already have kind of shared the races. We talk about East African races moving out into Central West Asia and maybe hitting South Asia and blowing around the world. The races we currently deal with in the Central Plains and New York State, you had stripe rust 2 years ago?
3 months ago, yeah.
Yeah, 3 years ago. 3 years in a row. Those races are actually races that came out of East Africa about 20 years ago. So we've already kind of had this sharing event. There's some differentiation in the races since. There's a couple distinct groups, but for the most part, I feel like what we do in Washington is directly relevant everywhere. And I know that because we actually send panels and populations to 3 locations in Ethiopia. So I feel like even more so than stem rust, it's, what we do in Washington can be beneficial around the world. So historically, historically this wasn't even there, but historically this is your stripe rust areas: California, Oregon, Washington, Pacific Northwest. Over the past 12–15 years, it's become a recurrent problem in the Central Plains. just some numbers for Washington specifically. In 2010, we had, and this is very conservative at a 7% of overall yield, we had about $57 million in losses in Washington State. I think that that number's wrong for a lot of reasons, and that doesn't include the cost of fungicides to protect 2 to 3 applications on a crop, on a wheat crop. That's kind of crazy. We don't apply fungicides 2, 3, 4 times on a wheat crop normally. But if we had more susceptible varieties, i.e. if programs in this region, my predecessors, had not been breeding for stripe rust resistance for the past 40 years, that would have probably been more like a $500 to $600 million loss in that 1 year in one state. So it's a big problem. So just, everybody knows this, I'll go really quick, but we basically, from my perspective as a breeder, I have 2 choices. I can go with, you know, what we call seedling or all stage or some sort of recognition-based resistance. In this case, YR5, a single gene that came from tetraploid wheat, was integrated into hexaploid wheat and is now being used globally. Like, there's only 2 or 3 genes being used around the world on purpose, and this is one of them. This is Avocet S. We currently don't have virulence to YR5, but all it takes is, you know, theoretically 1 mutation, especially if it's heterozygous in the rust, and we're susceptible, and that's where we have our boom and bust that we've been trying to get away from. In Washington, they've been really pioneers of adult plant resistance. Orville Vogel, Bob Allen started in the 1950s and 60s, even before Rajaram and some others in Simmet, in selecting for adult plant resistance to stripe rust. This is just an example of a hard red spring wheat I just released a couple years ago, which is actually now the #1 hard red spring wheat in the state. Typically, when we look at these pictures, we'd see under low temperature it's got more rust and high temperature less rust. This line happens to have kind of even a higher level of APR. But Alpawa here is a good example where the low temperature, it looks susceptible, and the high temperature it still looks susceptible to me, but supposedly that's adult plant resistance in that line. I should say I believe in both, you know, specific and non-specific resistance, but I'm trying to set up a story where stripe rust, I don't know which is which. And as a breeder, I cannot tell which is which. And this is example, real data from Shanming Chin, one of my close collaborators, who's a USDA ARS stripe rust pathologist. This is in 2010. You know, this is a seedling reaction to 2 races. These are just 2 races of stripe rust. 9 is the highest you can go, but 8 is basically fully susceptible. And these are 5 commercial varieties. And you can see all 5 of these varieties are seedling susceptible to both races. However, in adult plants with those same races, you can see we have lines that are either resistant to one as an adult plant and susceptible to the other, or resistant to the other and susceptible, or this kind of, what I think hopefully is more durable, resistant to both. But stripe rust doesn't seem to fit. You know, there's a few known genes for leaf rust and stem rust that are race-specific adult plant genes. They're expressed in the adult plant, but they've been defeated. Stripe rust, it seems like the majority of loci we're dealing with, and you could talk to Gene Milas at the University of Arkansas. They've really struggled with this in Kansas, Texas, Oklahoma, Nebraska the past couple of years. Is they think they have really good APR because they have seedling susceptibility and it's been resistant throughout its testing. And then the next year a race changes and those lines are susceptible. And so it's kind of hard to fit the leaf rust, stem rust model to stripe rust and really know where you're sitting. So I'm picking on Kels here. This was released a year before I arrived at WSU by my predecessor. It had been resistant with this, what they thought was really great adult plant resistance all throughout testing. Cross was probably made in 1999. And then it was under foundation seed multiplication in 2010, and this PST 127 race took over in the state, and Kels was dead before it was even challenged. It wasn't Kels's fault. It was just, you know, that gene in the background. Kels is still a good wheat. People chose to grow it. They still choose to grow it. They just spray fungicide, so it's not the end of the world, but it's not ideal when you're like, sure, this is it. It's got adult plant resistance. I'm there. And then the next year it's gone. So stripe rust is tricky. So this is my basic, you know, working logic for how I consider the 2 forms of resistance for stripe rust, and I'm just putting pros and cons. the con is adult plant resistance really is typically more temperature and phenologically sensitive, but they tend to break down less often. I just showed you a slide where that wasn't the case. And if it's truly, a different type of gene, I think they'll be durable forever. I think SR2 is a good example, LR34, some of these. But for striped rust, it's really hard to know which genes fit into that category without having the gene cloned. But, so I'm saying it's a con is that some are race specific, as I've just shown. Another con is we got to put a lot of genes together. For a breeder, that's not easy. I just had a discussion with Mark Searles yesterday about how frustrated he's been about one particular gene pyramiding project not getting done quite as fast as he wanted to. It's not easy when you get more than 3 or 4 or 5 genes you're trying to put together. Population sizes are hard. And I personally think we don't know anywhere near enough about the cost of some of these non-specific pleiotropic adult plant resistances. In my environment, we've developed isolines for LR34 and SR2 and a couple other adult plant resistance genes, and they consistently yield lower in Washington than their isogenic SIBs in nice yield trials. We have good APR genes in our backgrounds, but I think there's a lot unknown about modifiers, and so anyways. Another con is, and this is really important in places like Ethiopia, Kenya, Washington, Oregon, is the effect of this APR resistance on early season epidemic development and overwintering or oversummering if you're in a tropical area. If you're not resistant when you're small, but when you're resistant when you're big, there's still a lot of inoculum to deal with. And in Washington State, the environments, the nurseries I'll show you, it's not uncommon that I plant spring wheat and then it gets this big and it's infected in the field, naturally no induced epidemic, and it's infected until the day it heads or matures 4 or 5 months later. So you got to have some resistance early. In our yield trials where we have lines that have really strong seedling resistance and lines that have really, really strong adult plant resistance, they can still yield 20–30% different, which is a pretty big cost from that. That's not normal. You're not going to see that in New York. You're not going to see that in Kansas or some other areas where it really is based on the movement of an inoculum over later in the season. But in hot spots, in over summering or wintering areas, this is a problem. So I'm just going to flip it and say pretty much everything that I like about adult plant resistance, I don't like about the other, or vice versa. And so to me, it's all about keeping the wheat down here on this resistant line where we don't have yield loss and we have good resistance, we don't have new mutations. One thing I want to mention is there is a lot of noise. It still behaves as a quantitative trait in most of our studies. It's variable, there's G by E, and so it's not as straightforward as this graph might indicate in terms of the amount of severity or disease development based on genes over time. So this is what I come up with in the end, and this is how I live my life as a wheat breeder, which is I want several genes for both types of resistance. So basically my focus is how do I get that in a single line so that I have that early season protection, I have minimal inoculum production, I'm more durable, I have no yield loss. So what can we do? This kind of top part is what I tend to do, and the bottom part, the transgenics is not well, I guess I am a co-PI and a grant working on that too, but we can identify resistance loci. We can develop markers and allow people select. We can make genomic predictions, genomic selection. I'm really actually high on the potential of RNAi and transgenics for rust in particular, if we ever have transgenics in wheat, because that would solve a lot of problems as a breeder. Not getting into the politics of that, but it would be great for me. And then we develop germplasm, and so my job is to release superior varieties. So I'm talking a lot about rust, but that's, I could release a completely rust-susceptible line that's the top of the yield trial with good quality, has other disease and insect resistances, and my stakeholders would be happy. They'd say, yeah, you got us 10% more yield, that fungicide's cheap. they'll let me do that. Because in the U.S., that's a U.S. problem though. In Washington, or sorry, it's a Washington problem. Very highly productive area. So I have to take my sort of, philosophical approach to how I want durable resistance and what I want wheat to look like, and merge that with the very real expectations of royalties and stakeholders in terms of wanting as much yield as possible no matter what. And they can afford to do it with 3, 4, 5 fungicide applications, insecticide treatments, all these things. So now I'll get into the research. Just to basically everything I'm going to say, we rely on 3 to 4 primary locations for stripe rust testing, with the exception of some stuff going to East Africa. And that's Pullman, which is down here in southeastern Washington. We get stripe rust epidemics every year. It can be variable in terms of weather and moisture, but we have good screening nurseries. Central Ferry is just to the southwest of Pullman, and it sits down on the Snake River in an irrigated farm. It's only 20 miles away, but it's a month apart in terms of weather, and so we can plant there and actually stagger our nurseries, and it's warmer, so we start looking at G/E, temperature effects, things like that. Not directly, but at least we're capturing some of that variation. And then Mount Vernon, this is Skagit Valley, north of Seattle, about an hour. It's about a 6-hour drive for us, but that location's invaluable because stripe rust never dies there. It never goes anywhere. You plant, it's there. You harvest, it's there. It doesn't die. And so this is basically, extremely reliable. And then several studies. We also work with George Zubkovsky at UC Davis, and I'll explain what these regions might mean on the next slide. But for each of the studies I'm going to show, we get 2 to 3 years data, sometimes longer. We're measuring infection type, which is just it's from a fleck all the way up to a highly, healthy sporulating pustule on a 1 to 9 scale, and then just a Cobb scale for severity about how much leaf area is covered. And because our epidemics tend to be long-term, we can score them multiple times over the season, and for most traits we can calculate a area under the disease progress curve. Mount Vernon, that's true every single year no matter what. Sometimes Pullman, it's a one-time look. But we also, working with Shanming Chen, my staff do a tremendous amount of defined race seedling greenhouse assays as well. So what I mentioned about the regions on the previous slide, Mount Vernon's out here in what Shanming Chen calls Region 5, Pullman's in what's called Region 1, Davis is in Region 6, and this is his kind of loose areas of, or epidemiological zones of overwintering and inoculum sources for stripe rust. But the key point there is, like in Region 1, which is where Pullman sits, in Central Ferry, we do have multiple races present in the field. Generally, when we sample spores from our screening nurseries, we end up with 4 or 5 races that just naturally came in. And Central Ferry, they would be similar. And then Mount Vernon, there's different races, some older races, plus some sharing of races. But for the most part, the cascades do limit some of that crosstalk, so it's a little bit different race structure. In California, we can share races over the course of a decade, but year in and year out, they would tend to have a little bit different races than us. So we're trying to find, by screening, lines that are consistently resistant across different race populations in really different planting times, environmental conditions. So through the NEFA-funded wheat and barley triticity cap grant, there's a fairly large effort on leaf rust, stem rust, and stripe rust, and I'm the coordinator of that for the cap grant on the executive committee. And I'm just highlighting, and I'll describe a few that I'm going to talk about. One is we, through the cap grant, are screening the entire National Small Grains Germplasm Core collection, which is about 5,000 lines, 4,400 of them are genotyped by the grant, for seedling resistance in Washington to define races, as well as field resistance in Washington and in California for the spring wheats and winter, I'll just skip over that. But basically we have 9,000 SNPs on 4,400 lines, and we're phenotyping them in the greenhouse and in a lot of field locations. Some others include adapted elite germplasm. So in this case, there's 427 spring, it's a mixture of elite spring germplasm that's also been genotyped. We have other germplasm from soft red winter, more like Mark Sorrell's collaborators, other panels. So we got a lot of panels, a lot of phenotyping, genotyping going on. And then, not through the TCAP, but through other projects like Kebede Muleta's a Ethiopian PhD student of mine that's got a Beetle Borlog. We're screening a lot of Ethiopian tetraploid wheat, land races, and cultivars of hexaploid wheat. Through additional collaborations, additional Ethiopian durum wheats, wild tetraploids, and I'm trying to set up the story here where we basically can compare results across a lot of different panels because we have a lot of different diverse germplasm. So for the core collection itself, this is the NGSC Core. As I mentioned, the number here is about 5,000, but for whatever reason, 4,400 of them were genotyped and included. And it's a pretty good covering of breeding lines, cultivars, land races, and kind of unknown wheat germplasm collected over the past century from across the world. There's really not too heavy of an influence from any one particular geographic region. I think it's a pretty nice panel of lines to do this type of study on. So we've got a lot of research going on. On the next few slides, I'm just going to focus on the first 1,000 spring wheat accessions from that core collection. They're just a random sampling of the 3,000 total spring wheats, but we had enough seed of them a year earlier that we already have 3 years of data that I can present to you here. So that's why I'm cherry picking. So you guys are all smarter about this kind of stuff than I am, but you know, we've got a good diverse germplasm base, which kind of clearly makes sense based on the map I previously showed with germplasm from around the world. There's admixture, there's you know, definite population structure we can account for. This is just on those first 1,000 spring wheats. We get pretty reliable field data. Every once in a while we have a screening nursery that you know, the climate doesn't set up properly, but you can see there's highly significant correlations among the resistance phenotypes, whether we're screening it in this one region in Washington versus another one with a really different climate and different races. Or I could have added California to this. It's the same story. We tend to get really good phenotypic data. And we're dealing with a quite highly heritable trait as far as you know, these type of studies go. The heritabilities, this is for infection type, severity at just Pullman and Mount Vernon, and this is that 1,000 spring wheats. This is the winter wheat core collection, but whether it's winter or spring wheat, we're dealing with a trait we can phenotype pretty well. There are pretty significant genotype by year effects at times, particularly for severity. Infection type, infection type tends to be more stable, but severity is kind of that culmination of all the weather and everything. So this is where a breeder starts getting really frustrated. This is the GWAS hits or significant results from that first 1,000 spring wheats. This is 6 environments of California and Washington over the past 3 years, either transformed data or non-transformed. These are seedling resistance hits. And so you can start comparing, okay, I do have a seedling resistance gene that seems to be showing up in these environments. But for the most part, we have mainly genes that don't show up based on our GWAS analysis of seedling phenotypic data, which means we're basically dealing mostly with adult plant resistances, but they're also not always that consistent. So in this particular presentation, you know, it had to be present in 3 of the 6 environments. That effect had to be there in 3 of the 6 environments to make it onto this slide. But does that mean it's, you know, experimental error? Does that mean it's actually race specificity? Does that mean it's interaction with the environment? And the main point for somebody like me, though, is this list is like 110 things long. And for a wheat breeder that wants like maybe 5 genes, 5 great genes, that's all I want. What do I do with, how do I find them? How do I find them out of 110 things that have kind of variable? There are some really strong things. Maybe I really want that one that's most significant in red and it's consistent across environments. So maybe I should just look at that, but what if those have a fitness cost, or what if they have something else, or what if they're race specific and I don't know it? So I need somebody to tell me, wheat breeder, use these 5 things. And you know, some of them, these are actually, this data is phenological traits, heading date, plant height, and you can see some of those genes actually maybe consistently have something to do with the timing of the experiment, and we can account for those effects to some degree. So, from a research standpoint, I mean, I think we get decent results, and we take what was a fairly sparse map of really known effective stripe rust resistance genes or loci, and we start adding a lot of things to a map that people can say, oh yeah, that's there and it's real, but that still doesn't tell me what to use. And then we have things like this, which are SNPs that are in that Illumina chip that there is no genetic map position put to date, and what do I do with that? Maybe that was the best one. What if that was the most highly significant GWAS hit, but I don't even know where it is? Do I just use it? I'm basically trying to say I want Cornell to really develop genomic prediction and integrated genomic selection things, because I can't do this with what I'm doing now. So this is winter wheat, the same winter wheat panel over a couple years, and these are different. I'm not going to compare them, but pretty much these are different in the winter wheat germplasm than they were in the spring wheat germplasm. And I don't think it's an artifact. We've got really complex resistance to stripe rust. It's not as simple in a lot of ways as stem rust or leaf rust. So then we add seedling data on top of that. In some cases, there's beautiful results, and the same collection of 4,400 lines we've run with 5 races of stem rust, including the East African races, and sometimes you just get these perfect hits where the p-value is phenomenal, and it's basically diagnostic. I can look in 5,000 lines across the world. If it has that marker, it has that phenotype, and that's good. But this is rare, having that kind of significant diagnostic marker, that the one marker itself is the haplotype, and you're pretty much done. You've got your breeder-friendly marker. For the most part, we get more stuff along these lines where we get some hits there, some hits there. The allele, or the haplotype, is difficult to discern. We have maybe a fairly rare allele, but not every line is resistant with that allele. So then you have to kind of scan out and start looking for the right haplotypes. And for an impatient wheat breeder, it's complicated. So that's kind of hexaploid. We've applied some of this to tetraploid wheats as well. In this case, there's 178 cultivated Emmer wheat accessions that we phenotyped in the field and greenhouse over 3 years using the same Illumina 9K, and analyze it as many different ways as Cornell can publish, and see what to do. These are actually more accessible for me. In the case of this association mapping in Emmer wheat, we have 2 loci that are strong and consistent that show up, one of them in the seedling and in the field, one of them just in the field, and I go, yeah, I can use that one thing, and it's really good and strong, and move forward with it. But it's in Emmer wheat, so I now got to spend 5 years or 6 years making a better parent out of it first, which is a problem. And this is, just more kind of struggles or frustration. This is that data set analyzed with either using tassel only, or tassel with ridge regression BLUP, or tassel with GAPIT, or some combination. And as an impatient wheat breeder, I don't know which one of these to use. I mean, I like the yellow ones that everything identifies, but then we come to results like this, where my experience to date is anytime there's more than a few centimorgans in genetic position between significant hits, they tend to be different loci. So if we're seeing 11 centimorgans, that could be 2, but do I have to publish another paper, or do a test cross or what to figure out if those are 2 things or 1 thing? It's a challenge. And then, what do we do with these where, tassel and our BLUP are identifying, consistent effects that seems to, map to a defined position? It's probably a real resistance locus that we should be considering, but it didn't show up in GAPIT or something, I don't know. And then for me, and I'm glad I had the opportunity to visit with Jean-Luc this morning, for me, the immediate thing is go to your accessible elite germplasm and do this so you can immediately get going. So we had a kind of a ad hoc contributions and funding to Genotype, 427 sort of historic cultivars, elite breeding lines, major varieties from Montana, Washington, or Idaho, Oregon, California. But they're made up of several market classes. We grow soft white spring wheat, hard white spring wheat, hard red, and even some spring club wheat, which would be in this germplasm that doesn't show up. So we do that. and we phenotype it and genotype it, and we come up with one locus on 1B. This germplasm has been selected for stripe rust resistance for 60 years. We have very good adult plant resistance. We have some seedling or race-specific resistance. And all I get is one locus. You know, what happened? I know there's more genes in there than that. And Jean-Luc pretty much explained that to me this morning, which is the resistance in our soft white spring wheats would be largely different genes than in our hard reds. And when combining, and that's the theory we'll need to test, but when combining all these into one association mapping, basically the structure analysis is pulling out all those relationships and basically killing every other potentially significant resistance locus in the group. This is YR15. It was probably present in 10 out of the 427 accessions. It's a bulletproof gene, nice high level of resistance. Come up with a diagnostic marker, it's great. There's genotyping labs across the country already using this, but I already knew how to use that gene. I wanted to know what was behind, you know, what else was in there that I could use. Yeah, that's for this panel has diverse and a lot of adult plant resistance to stripe rust, and all I come up with is 2 little measly loci compared to the big core collection. So, you know, we'll chip away at how to do it, but in hindsight, we should have never, like Jean-Luc told me, we shouldn't be, you know, using different populations all as one population and expecting a different result. So it would have been smarter to have 300 or 400 just soft whites and not waste money by spreading it across that. So I don't have any conclusions, but I do have some thoughts or complaints, and I think there's too many opportunities, and a big question for me is how do we prioritize resistance loci? It's really easy to go into a genome-wide association panel and identify things, but we don't have performance data on a 1,000 winter core or 3,000 core collections accessions. So determining linkage relationship, is that linked to poor quality? Is that linked to this and that? We can't really do that in those core collections. It's too expensive. So that's, you know, interactions. Mark Sorrells and others have been doing nice work on interactions of adult plant resistance and stem rust, and I think there's a big need in stripe rust for that same kind of work. What kind of fitness costs with some of these APR? I believe there are fitness costs with many of them. Some examples are published very nicely. How do I know which ones are going to be more durable? And then, you know, what are we missing? And this goes back to like my elite spring wheat germplasm example. I know I'm missing at least 5 genes I can name and I have a good marker for, but that's my fault for having a bad population design. But still, in general, this fixed of masking or power or, you know, limitations on minor allele frequency. In the wheat chips to date, we do have an issue. I didn't really mention it all throughout, but the D genome is particularly devoid of markers, and I think there are more resistance genes on the D genome than we're detecting. We just don't have the density to detect that, so I'm hoping that GBS and other approaches applied to some of these same studies will help. The issue of ascertainment bias in the marker platforms themselves. This one is a real challenge for me, is how do I identify lines that have each resistance locus for sure? To go through and make a bunch of backcross, isogenic lines using subtly different haplotypes that all had the good marker or the good 2 markers, but different haplotypes fairly close, that's a challenge, and I need to know what lines to use. And if there's multiple genes in each line, which in most cases there are, you don't know. You can't simply look at the phenotypic data. And so, I've become more and more convinced that, predicting and using genomic selection methods is going to be the only way I can make, sense of this in this world of trying to put everything together to hopefully realize durability. So with that, I'm going to thank Shenming Chen, who's an amazing collaborator, who's basically willing to do everything all the time. We screen about 20,000 lines a year routinely for stripe rust resistance, equally, but he's never said no yet. Stephen Zhu, who's a cytogeneticist and wheat germplasm enhancement person at USDA Fargo, North Dakota, a great collaborator. George Zubkovsky is involved in the TCAP work on stripe rust. Roberto Tuberosa at the University of Bologna on on some of the Durham Tetraploid Wheat Association mapping projects, and then Bekele Abayo and Badata Gurma at EIR, who work on the Beecher Borlaug project as well as on the DRW project, but have facilitated screening for us in East Africa. Xiaoman Chow and the other genotyping USDA labs for a lot of help on these, and Mike Bondman at the National Small Grains Germplasm Collection. And I didn't talk about stem rust, but pretty much all of my interactions and things I do with people, I think has arisen a lot from involvement with these same people and resources through the DRW project led here at Cornell. So with that, take any questions.